Welcome to extraction of RNA from laser capture microdissected clinical FFPE samples presented by BioCompare and sponsored by Covaris. My name is Tamlin Oliver, Managing Editor of BioCompare, and I'll be the moderator for this webinar. Today, Enni Markkanen will talk about the RNA extraction workflow her team developed to analyze gene expression changes in small subsections of cancer tumor tissue isolated by laser capture microdissection. She'll be joined by Michael Hanig, European Sales Manager, as well as a workflow specialist at Covaris. They will get started shortly, but first I want to go over a few housekeeping items. We'll hold a live Q&A session at the end of the presentations. You can submit a question for the Q&A session at any time using the Ask a Question tab in the upper right corner of your screen. The right side also features an overview of the webinar as well as more information on the speakers. In addition, your console is fully customizable, and we encourage you to move, maximize, or minimize any of the tools on your screen to optimize your viewing experience. If you have technical difficulties at any time, please click on the Test Your Connection button at the bottom of your screen. From there, you can access webinar support. We've also provided social media widgets beneath the webinar window to facilitate the sharing of this webinar with colleagues. All right, with all that done, I can introduce our first speaker, Michael Hanig. Michael is European Sales Manager for Covaris. He has extensive experience supporting scientists on their workflows, including proteomics, cell lysis, chromatin shearing, and NGS applications. Before joining Covaris, Michael worked at LGC Genomics. He received his PhD in microbiology and marine biology from the University of Rostock and Leibniz Institute of Baltic Research. He worked for 3 years at the Max Planck Institute for Terrestrial Microbiology Marburg as a postdoc. Michael will speak first with a brief overview and then introduce Enni, who will discuss her research and new workflow. Welcome to both of you. Michael, please get this webinar started.
Thanks, Tamlin, for the introduction. Hello, everyone, and welcome to our FFPE-related webinar developed in collaboration with the University of Zurich. Today's webinar is brought to you by BioCompare and sponsored by Covaris. I'm Michael Hanig, European Sales Manager at Covaris. After a quick introduction to the Covaris technology, I will hand over to Dr. Enni Markkanen, who will present us the extraction of RNA from laser capture microdissected clinical FFPE samples. As this is interactive, please submit all your questions and comments during the presentation, as we are happy to answer them later in the Q&A session. Let's start with a brief introduction to Covaris technology. Covaris provides tools and technologies to improve the pre-analytical sample preparation. 22 years ago, we developed a technology called AFA, Adaptive Focused Acoustics, that can be used for multiple applications. Covaris is well known as the gold standard for DNA shearing, and with more than 6,000 instruments installed worldwide, we are present in almost all main genomic centers. In addition to genomics applications, our instruments can standardize the extraction and preparation of other biomolecules, such as chromatin, proteins, metabolites, and much more. Depending on the requirements of the lab, we offer solutions for low to high throughput demands. AFA technology is computer controlled and tunable. With the release of the focused ultrasonic acoustics, we operate outside the human audible range in comparison to normal sonication. This video illustrates how AFA induces cavitation in the sample. Cavitation bubbles will appear, grow, and finally collapse, thus releasing a shearing force to the sample. Altogether, this allows us to maintain isothermal conditions in the sample and to provide reproducible results. Covaris AFA can be used in different steps of your methods, starting from various sample types, including formalin-fixed paraffin-embedded material. Depending on the type of biomolecule of interest and the downstream analysis, AFA can be not only used for the lysis of cells, but also for multiple steps in more complex workflows. Especially for FFPE samples, the release of biomolecules is quite difficult, especially if you are dealing with very small or old samples and low-quality samples. You can see here the emulsification of an FFPE sample in an aqueous solution. AFA actively removes the paraffin from samples by breaking it down into small fragments, as well as rehydrates the tissue. Kouvaris offers solutions to co-extract RNA and DNA from the same sample, and the results for the RNA extraction will be presented by Enni in her talk. Now I'm happy to introduce Dr. Enni Markkanen. She's working at the University of Zurich at the Institute of Veterinary Pharmacology and Toxicology. Enni started her scientific career in Zurich before she spent 2 years at the University of Oxford as a postdoc. In 2015, she became a group leader at the University of Zurich. I will now hand over to Enni, and I'm looking forward to see your interesting presentation.
Thanks, Michael, for the introduction. In the next 15 minutes or so, I would like to share with you how we are using extraction of RNA from laser capture microdissected clinical FFPE samples to help our research. The main focus of my lab is centered on understanding tumor biology, and more precisely, we want to better understand the interaction of tumor cells with the surrounding stroma. In addition to cancer cells, tumors contain an assembly of many different cell types, such as fibroblasts, which are the spindle-shaped cells, the reddish ones, or the dark red ones in this picture, or immune cells, vascular cells, adipocytes, and others. Classically, cancer cells have been considered as the only bad guys in this cell mix. More recently, however, it has become clear that cancer cells heavily depend on the surrounding stroma. The interaction between neoplastic cells and the surrounding stroma strongly influences tumor development. This stroma that is found so close to and within tumors is usually referred to as cancer-associated stroma. To analyze what is going on in the tumor stroma, one of the models we are using is breast cancer in dogs. By many, the dog is considered a good model for human breast cancer. Breast cancer is the most frequent cancer among women and female dogs. Similarities and also advantages over rodent models include cancer developing spontaneously. There are very similar clinics and pathophysiology between the two species. There are many aberrations in the equivalent genes as in human breast cancer, for instance, TP53, BRCA1, BRCA2, just to name a few. Dogs very often closely share the environment with their owners, so they get exposed to the same environmental factors as we do. They show expanded lifespan compared to rodent models, and they receive a high-level health care and cancer treatment, which makes it possible to get samples for analysis. When we started to consider using the dog as a model for our studies, we then wondered, well, what about the cancer-associated stroma? We went into literature but could find virtually nothing on the subject. We then asked, how can you possibly claim that the dog is a good model for human breast cancer if you don't even know what half of the picture looks like? And that's exactly the question we've been analyzing over the last few years. But how can you specifically analyze the stromal component in clinical tumor samples? You could use data from bulk tumor sequencing, such as from TCGA and other resources. The problem, however, is that with bulk tumor sequencing, as the name suggests, the whole tumor is mashed up, and the sequencing gives you an average of all different cells that were present in the tumor at the time. It's a bit like taking a fruit bowl, putting all the fruit into a blender, and then getting a nice smoothie out of it. When you look only at the smoothie in the end, it will be difficult to say what the banana looked like that went into the blender, or exactly how many grapes were there to start with. And that is exactly the problem you're facing when you're trying to extrapolate data regarding tumor stroma from bulk tumor sequencing results. So to analyze cancer-associated stroma in clinical samples, you should ideally be able to isolate it somehow. To do this, we use so-called laser capture microdissection. This technique allows you to use thin sections of formalin-fixed paraffin-embedded tissue from clinical samples to define your areas of interest under the microscope and to cut out exactly these areas of interest so you can extract, for instance, RNA or protein for further analysis. This slide shows you the normal workflow we're using. First, you choose the cases, after which there is a slide preparation and tissue staining step that is followed by laser capture microdissection. You can see an example of laser capture microdissect tissue on the top right of the slide, where I'm holding a cap containing isolated tissue. Then you isolate RNA and perform quality control before ultimately analyzing the RNA. Often, this is done by qPCR. But with qPCR, you can only see what you want to see because you have to define your targets of interest beforehand. Also, the number of targets you can look at is rather limited, for instance, by sample size, especially in this approach that we're using. A better option to get a good overview of what is happening with gene expression in the samples would be to use RNA-seq. So we were wondering, can we analyze these small pieces of FFPE tissue using RNA-seq? FFPE is a vast and valuable resource of patient material which is lying around in basically every pathology lab in the entire world. It entails a large amount of clinically relevant data, and nucleic acids can be recovered decades after fixation. There is a drawback, however, which is that chemical processing during the formalin fixation and paraffin embedding severely impacts on RNA quality. So when we asked literature about RNA-seq of small deparaffinized stained and microdissected FFPE tissue, back then it basically said, no way. We said, well, let's see about that. During initial experiments, one of the main limitations we encountered was that we only got very small amounts of RNA out of our samples when we were following the conventional RNA isolation protocol that is based on tissue digestion with proteinase K only, which is what you can see on the left-hand side of the screen. Regardless of how hard we tried to digest, we always ended up having macroscopically visible tissue pieces and very low amounts of RNA that we could get out. Because we had those big tissue pieces that we could see by eye, we thought there should be room for some improvement. And indeed there was. We then came across the Kouvaris focused-ultrasonication protocol for RNA isolation from formalin-fixed paraffin-embedded tissue, which you can see on the right-hand side of the screen. And we decided to give it a go. So we isolated CAS and normal stroma from 13 clinical samples of canine mammary tumors, comparing the conventional RNA extraction with the new Covaris-based protocol. You can see on the very left graph that the new or Covaris-based protocol yielded on average between 8 to 12-fold more RNA compared to the conventional protease-only based old protocol. In the middle panel, you can see that the RNA quality was not really altered using the Kouvaris ultrasonication protocol. And finally, we saw that we could get sequencing data from samples processed in this way. So using Kouvaris ultrasonication, we achieved the development of a protocol for RNA extraction after laser capture microdissection of formalin-fixed paraffin-embedded tissue, which is amenable to RNA sequencing. We then went ahead and isolated cancer-associated stroma matched normal stroma from a cohort of 14 simple canine mammary carcinoma and sequenced them. On the top left in the PCA, you can see two different clusters. One that is on the left and contains only samples from normal stroma in black, and the one that is on the right and contains only red dots from cancer-associated stromal samples. So the distinction between the normal stroma and the cancer-associated stroma is the major source of variability in the data. You can also appreciate the strong difference between gene expression in the normal samples and cancer-associated stromal samples in the heat map, which is on the right of your screen. These results clearly showed that there is strong reprogramming of the stroma in canine mammary carcinoma. Next, we asked how the canine cancer-associated stroma compares to that from human breast cancers. To analyze this, we compared our data to a published human dataset of stroma from breast cancer that was similar to ours, but was obtained using microarray. We did this using two methods, self-contained gene set testing on the left and competitive gene set enrichment analysis on the right. Both of these showed that there are strong correlations in expression changes between cancer-associated stroma from canine and human breast cancer. We then wanted to compare our data to a larger data set to further validate these results. To do this, we used the TCGA's entire breast cancer dataset, which comprises more than 1,300 tumors that have been sequenced. Importantly, however, these are data from bulk tumor sequencing. So as I told you earlier, these represent a mix of tumor and stromal cells. Therefore, we first had to rank these tumors according to their stromal content, using a stromal signature called estimate. You can see this along the x-axis. So the tumors on the left are the ones with very little stroma, and the ones on the right contain a lot of stroma. When we now analyze enrichment of our canine-derived stromal signature on the y-axis in these samples, we can clearly see a positive correlation between the canine-derived stromal signature and the stromal content of human tumors. This further strengthens the notion that canine and human tumor stroma is very similar indeed. To quickly sum up, we find evidence of strong stromal reprogramming in canine mammary tumors, and there is a high grade of molecular homology between cancer-associated stroma from canine and human breast cancer samples. So I told you earlier that the stroma can strongly influence malignancy and progression of a tumor. So far, we had only analyzed what differentiates malignant tumors or carcinomas, which you can see in the bottom, from normal stroma, which you can see at the top of the slide. But there is a benign intermediate between these two, which are the so-called adenomas. Interestingly enough, there is no data about stromal reprogramming in human or other spontaneously derived benign mammary adenomas in literature. Therefore, we then wondered, does stroma in adenomas undergo any reprogramming? And if it does, are there differences in reprogramming between benign and malignant tumors? To answer these questions, we isolated calves and normal stroma from 13 benign mammary adenomas. Again, we saw that the gene expression in normal stroma was very different from that in calves, which you can see on the left in this PCA. So clearly there is stromal remodeling going on even in benign adenomas. We then wondered how the remodeling between canine adenoma and carcinoma compared, so between the malignant and the benign tumors. Here in this PCA, you can see that while the normal stroma from both datasets behaves very similarly, those are the pale blue and the dark blue dots, calves in adenoma, which are the orange dots, clearly cluster separately from calves in carcinoma, the red dots on the left. This suggests that there are substantial differences in stromal remodeling between adenomas and carcinomas. To understand what exactly is different between the stroma of benign and malignant tumors, we then identified the genes that were most discriminatory between normal stroma, the stroma of adenoma, and the stroma of carcinoma. You can see the top 40 genes in this heat map that discriminate normal stroma and the other two from each other. There are a few interesting things that can be seen here. For instance, these are the genes that are highly similarly expressing in normal stroma and stroma from the benign tumors, the adenomas, but they're highly upregulated in the stroma from carcinoma. These are the genes which are specifically downregulated only in the stroma from carcinoma. These are the genes which are specifically downregulated only in the benign tumors. And finally, these are the genes which are specifically upregulated in the benign tumors and not really changing between normal stroma and malignant tumors. So if you think about it, these are the genes whose expression you'd expect to be beneficial in the sense that you get more benign tumors if they're highly expressed. We then went on to check the influence of exactly the expression level of these genes on the cumulative survival of the TCGA breast cancer dataset. Interestingly enough, when we divided the tumors into low expressing and high expressing with the cutoff set at 50%, the tumors with high expression of 7 of these 10 genes in this cluster showed significantly better patient survival than the tumors that had low expression of these targets. Therefore, these results suggest that differential stromal remodeling between benign and malignant tumors can identify potential disease-modulating components in both human as well as canine mammary carcinomas. So, to sum up, we have demonstrated that different cancer-associated stromal signatures in various stages of malignancy exist and have identified a set of disease-modulating components whose function should be studied in further detail. Here, I would like to take the opportunity to say a few words about the sample requirements for our workflow. Sample size often seems like the obvious limiting factor, but this example of adenomas demonstrates that it is possible to get good data out of tiny tissue pieces. In adenoma, it is very difficult to find large pieces of cancer-associated stroma. This is a case where we could only isolate that tiny little scrap you can see here on the right. We isolated this small piece from 2 consecutive sections of tissue and went on to successfully isolate enough RNA for RNA sequencing. One big question that usually comes up is that about RNA quality from FFPE tissue. This table is just to show you the quality of RNA that we routinely get when isolating cancer-associated stroma from clinical samples. On the left, you can see data for carcinomas, on the right for adenomas. For RNA from FFPE, it is best to determine RNA quality using the so-called DV200 value, which estimates what proportion of RNA is longer than 200 nucleotides. You can see in the last column of both tables that the DV200 values are usually somewhere between 65 and 80%, which is considered quite good using this Covaris-based approach. Another worry is usually sample age. There are several reports in literature that RNA quality and quantity strongly decreases with increasing sample age, and some of these reports even suggest not to use tissue older than 2 years or so. In this table, you can see in the last column that a substantial number of these samples that we used in a yet unpublished study were more than 10 years old, so more than 120 months. We could not see any problem with these samples, nor did they perform worse than the fresher samples. Thus, in our hands at least, sample age does not seem to play a major role, provided the samples have been stored under good conditions. So let me come to the end and quickly reiterate some of the important points we touched upon today. I hope I could convince you that the Covaris-based protocol for RNA extraction enables analysis of laser capture microdissected subsections of formalin-fixed paraffin-embedded tissue by RNA-seq. Our data demonstrates massive stromal reprogramming in canine simple mammary carcinomas and also adenomas. There is extensive molecular homology between cancer-associated stroma from human and canine mammary carcinoma, which support the validity of spontaneous canine breast cancer as a model for the human disease. And importantly, laser capture microdissection of FFPE tissue enables detailed molecular analysis of specific areas of interest from any archival tissue samples of a wide variety of diseases and organisms. Finally, I'd like to end with my acknowledgement slide, and I would like to thank the people in my lab who've been really instrumental in establishing all this workflow and getting all this data. And also our bioinformatician, Sina Nasiri, who is extremely instrumental in really getting the data interpreted in the correct way. And also the funding agencies for their support of our work. And with that, I'd like to close my presentation, and I'm hoping to get several questions from you. Thank you.
Thank you, Michael and Enni, for those informative presentations. We'll start our live Q&A in just a minute, but first I have some polling questions. Okay, our first question is, how are you preparing your DNA and RNA from FFPE samples? extraction using organic solvents, commercial kit, Covaris FFPE emulsification, or other? I'm going to give you a couple seconds to answer that. Okay, and the results should be displaying on your screen right now. Yep, it seems overwhelmingly, or not overwhelmingly, but the commercial kits seem to be most popular. All right, let's go to the second poll question. Do you have enough high-quality DNA or RNA for analysis? No, yes, or I need to improve both quality and yield. I'm going to give you a couple seconds to answer that. All right, it seems a lot of people need to improve quality and yield, or maybe just a couple that answered, so maybe that's not entirely representative. All right, let's go on to the next one. Do you have access to a Kouvaris AFA-ultrasonicator at your institution? Yes, no, or I would like to get one. I'm going to give you a couple seconds on that. While we're waiting for these results, you can go ahead and submit any questions you might have for Michael or Enni. They're ready. They're standing by, ready to answer your questions. All right, let's just get these final poll results. All right, so a lot of people don't have access to one, but a considerable percentage would like to have access to one. All right, thank you for taking the time to answer those poll questions. Michael and Enni, are you ready for answering questions?
Absolutely, I am.
Yes, absolutely.
Great. All right, Michael, the first question is for you. Can you extract DNA and RNA from the same sample?
Yes, Tamlin. We can extract DNA and RNA from the same sample. So we have a kit that is called truXTRAC, and there we have an extraction method where we first extract the RNA, and then later we can extract the DNA. So we clean both RNA and DNA then in two separate ways. And it's nice because, of course, if you have limited amount of FFPE material, you like to do that from the same sample, and that's possible.
Okay, thank you, Michael. Enni, you're up next. How do RNA-seq results from FFPE compare to those obtained from fresh frozen tissue?
So thanks for the question, Tammy. I have to admit we haven't done really any extensive comparisons between fresh frozen and FFPE tissue, because we mainly work with FFPE tissue. But there are several publications in literature which have done, at least from entire sections of FFPE or fresh frozen material, they've compared RNA sequencing results. So taking quite a lot of material, actually. But there the correlations are very, very good. So if I remember correctly, the correlation is higher than 0.9. So really high correlation, suggesting that you really get very, very similar results taking either type of tissue.
Okay, thank you for that. Michael, we're back to you. What is exactly the benefit of AFA for the FFPE extraction?
Yeah, thanks for the question. The benefit, what you also have seen in my introduction, is really the deparaffinization of an FFPE sample, and within an LCM sample, of course, it's the additional benefit that we rehydrate the tissue also and make it then later accessible for the digestion. And together with the deparaffinization that is actively done by the treatment of AFA and the rehydration, we get the RNA and DNA better out of the tissue and of the sample. So this often leads then to higher results in quality and quantity.
Okay. Thank you. Enni, the next question is yours. Is this method restricted to analysis of stroma?
Absolutely not. So you can really think of analysis of any kind of tissue which which includes RNA or DNA. So whenever you can isolate that piece of tissue from FFPE, or you can take that piece of tissue and isolate nucleic acids from it, you can use it.
Okay, thank you. Back to you, Michael. What else can you extract from FFPE samples?
So actually, we have developed different kits for DNA and RNA from FFPE samples. So combined and purely RNA and DNA. And in addition, we have workflows established for proteins from FFPE and also for chromatin from FFPE samples. So we have different versions that can be then used.
Okay, thank you. Enni, I'm assuming the next question is for you. If not, Michael, you can jump in. Which company RNA-seq library kit was used?
So this is right now I'm struggling to find the word. It's the Clontech, yes, Clontech Pico input mammalian RNA sequencing product. So this really allows you to use extremely low amounts of RNA. So we routinely submit 4 nanograms of total RNA for sequencing. And that's enough per round.
Okay, thank you. I have another question for you. It's a multi-part one. So you extract from 2 consecutive sections of 3 microns, 5 microns, and 10 microns. What's the final RNA quantity obtained, and what RNA input in NGS experiment? And also, what is the sample prep?
So we routinely use 10 micron sections. In the beginning, when we started setting up this workflow, we tried, of course, different section thicknesses. And the thing was, if the section is too thin, you really struggle to get enough material because you get really low amounts of material anyway, as you saw in the presentation. And if the sections are too thick, you cannot really discriminate anything anymore under the microscope. So in our experience, really 10 micron is the optimal thickness to be able to see what you're isolating and also to get the nicest yields of RNA out of your sections. The sample preparation, like the method of how to prepare the samples for laser capture microdissection, you mean? So what we do is we just cut, it's a routine pathology lab procedure. You take your formalin-fixed paraffin-embedded tissue samples and you cut 10 micron sections. You have to be very careful if you isolate RNA that you're using everything is absolutely clean that you're using. So you're using RNase-free water. You really have to use clean blades to cut the sections. Otherwise, you might contaminate the sections with RNases, which then, of course, does not help you in the downstream applications. But this is really, if you have a pathology lab at your disposal and you give them the specifics, and you can find all of the specifics in the papers that were mentioned in the presentation, really in the materials and methods section, this is really not something extremely difficult. We have had several pathology labs, depending on where the samples come from, performing this workflow for us, and it has never been a problem.
Okay, here's another question that asks about the thickness of your FFPE sections and what is the size in micron length and width for RNA-seq? And then do you notice any issues of HE stained tissue for input, or would you prefer to use unstained tissue?
So great questions. Those are all things that we had to test ourselves when we started. So as I said before, generally we use 10 micron sections because we just found that to be an optimal thickness, at least for the type of tissue we're using. It might be that if you're using, more higher cellular cell containing tissues, you might be able to go to a slightly thinner thickness, maybe to 8 microns or so. But usually 10 microns is really also good to still see tissue architecture, tissue morphology quite nicely under the microscope. So to the next question regarding what length and width of tissue size that you excise, we have never really measured because it so depends on your sample. In some tumors, for us, because we're isolating the stroma, in some tumors, it's extremely simple to find big chunks of stroma, which you can just isolate, in one go and you're done very, very quickly with isolation from that sample. In others, you really have to go and pick together tiny pieces from here and there, and it's just much more work intense and takes much longer. So we never really measured any of the surface area, if you want, that we isolate. But as I showed you with this adenoma sample, these are tiny pieces that you can isolate from 2 consecutive sections. In that case, most of the time, you really get enough RNA out for the specialized workflow afterwards for sequencing of under 10 nanograms of RNA.
Okay, thank you. We're going to move over to Mike.
Okay, sorry.
Enni, I'm sorry. Did I cut you off? Was there another?
No. I think there was a third part to the question which I again forgot. So I wanted to ask you.
Was it about HE staining?
Yes, H&E staining. Yes, that's something. So that's something we tried out first. We have to stain our samples because otherwise you cannot reliably differentiate between stroma and between tumor cells or anything really you want to isolate in LCM. So we do necessitate staining of samples. And in the beginning, we compared H&E, Cresol Violet, and 2 or 3 other methods of tissue staining. And what we figured out, or in our hands at least for stroma, Cresol Violet seemed to work absolutely best also in terms of RNA quantity and quality that you could get out of the samples. Now, what we always do routinely is if we have our consecutive sections being prepared, the first section is always is a thinner one, which is always stained by H&E. So you can exactly look at the tissue, really seeing the perfect morphology using H&E, looking at that, and then you go to the next section with Cresol Violet and you know really where you are. So I think that's very important to really know what you're isolating at any given time. That's also why it's very important, I think, to include very well-trained pathologists in any of these workflows. Otherwise, you might end up isolating things which are just not what you would like to do.
All right, Enni, we're going to give you a break and go to Michael, and then I think I have 4 or 5 more questions for you. So, Michael, do you have an automated extraction method?
Yes, Tamlin, we have an automated extraction method. Normally people start with our kits that are column-based, and then they do it manually. And then in the end, if they ramp up, if they increase their throughput, we also have kits with beads that you can automate. So that can be done then on a Kingfisher system, for example, or on a liquid handler. So yes, we can automate this process, and even the column-based methods were automated by some customers.
Okay, thank you. Enni, back to you. You said that the results were good for FFPE tissue that was stored under good conditions for more than 10 years. What are those good conditions?
I think good conditions in that case, because FFPE usually is stored at room temperature. Right, that's why it's also so widely used, because it's so easy to store. So I think in that case it's very important that you don't go between 40 degrees and 5 degrees. You don't have huge temperature changes all the time. And the other thing is low humidity, that the blocks don't get exposed to humid environments. Other than that, honestly, maybe no direct sunlight, but usually in any archive you don't even have windows, so I wouldn't expect sunlight to get into any archive that contains tissue samples. So really, if you want, in quotation marks, normal circumstances, really should do.
Okay, Enni, have you analyzed human tissues using this method?
Yes, we have. We have several collaborations with different hospitals and research groups from human hospitals, and we have analyzed, tumor cells. We have analyzed tumor stroma coming from human samples. We have also analyzed other types of diseases, such as psoriasis-like diseases, and it really. Actually, if you're using human samples, it's a bit easier from the point of view that if you get your RNA sequencing results, you obviously have much better databases to look for to align your sequencing results to. So you get even better data out of it when you're using human or probably also mouse samples, which we haven't. Oh, yes, we have done that as well. True. So yes, the dog it brings in an additional complication in terms of the databases, the genome annotation and everything not being so ideal as for human samples, for instance.
Great. Just a few more questions for you, and then we're going to have to wrap this up. How many subsequent 10-micron sections did you use for one lesion?
It very much depends on how big the section or how much tissue of interest is in every section. We routinely my students, they have 3 to 4 consecutive sections prepared. Usually they can even fit 2 sections onto one of these slides that we use for LCM. So you prepare 2 slides with 3 to 4 sections. Sometimes after, isolation from 2 of the sections, you end up already having so much material, and at some point you have that a bit in your fingertips, what is enough and what is not enough, that they just stop after isolation from 2 sections. Sometimes it really goes up to 4 sections, but rarely do we need more than that.
Okay, the next question is, what tissue sections do you use? Can you use the tissue sections cut 6 months ago that are stored at 4 degrees?
We have not tested that. I know that there are some ideas around in literature that it's not ideal. I would still probably give it a go. You will see very quickly after the RNA isolation, when you do the QC, you will see immediately if you have a substantial decrease compared to the canonically processed samples with regards to RNA quality. But I cannot really tell you firsthand from experience whether it does influence or it doesn't.
Michael, we actually do have one more question for you. So as long as you're still there, how long is the extraction method?
Yes, I'm here. The extraction method, how long it is, this depends a bit on, of course, the kit itself. So if you want to extract RNA and DNA, or only RNA or only DNA, but the point is that we do not need to have overnight digestions, because we can already actively remove the paraffin and then decrease the time for the proteinase K digestion and the decross-linking. So that means, overall, we can do that in some hours. And then, of course, it depends on the number of samples and if you do everything automatic in an automatic way, or if you do that manually. But within some hours, you can just extract both.
Have you heard of a research group that has used this technique to do RNA-seq on organisms that have infested the skin, like leishmaniasis? I know I said that wrong. I'm sorry.
Leishmaniasis. I think it's a parasite which lives in the skin. I'm not aware right now of any research group having done that, even though probably there is out there someone who has done it, because I think that's exactly a good thing to do, or a good technique to use for those kind of questions. So I really don't see why it shouldn't work. You could isolate the parasite separately, and then the surrounding tissue, the surrounding dermis, for instance, and look at what kind of reaction you get in the dermis, and then you could, for instance, isolate some healthy dermis from farther away. That would be a very interesting thing to do, I think, and I really don't see a reason why this shouldn't work.
Okay, thank you for that. And Thank you, Michael and Enni, for joining us today, and also a special thank you to Covaris for sponsoring today's event. Our next event is on July 30th. It's also sponsored by Covaris, and it discusses a high-throughput method for high-quality, high-molecular-weight DNA extraction from tissues. You can get more information about that webinar or register on our site, biocompare.com, but the link and more information is also available in the webinar overview module for today's event. So we hope to see you there. One final thing, this webinar will be available on demand in about 24 hours. You'll get an email advising you of that. You can watch it again or share it with colleagues. And we thank you very much for joining us today, and we look forward to seeing you at our next event on July 30th. Thank you.